Hi class, in this recording we're going to focus primarily on the citric acid cycle, also known as the Krebs cycle, also known as the tricarboxylic acid cycle. It really depends on what your background is. If you're pretty crusty and you've been trained a while ago, you were probably taught the Krebs cycle. So I'm thinking back to my high school biology teacher. He was a Krebs cycle kind of guy. My cell biology and molecular biology professors when I was in college, they were more citric acid cycle kind of people, and then my biochemistry profs and my organic chemistry profs. They were very excited about using the phrase tricarboxylic acid cycle, but they're all referring to the same thing. For me in particular, I'm more of a citric acid cycle kind of guy, just for the sake of full disclosure. Now, let's review. We're referring to aerobic respiration. We have lots of oxygen present. So during aerobic respiration, glycolysis will take one 6-carbon glucose, and break it down into two 3-carbon pyruvic acids. So the one glucose gets turned into 2 pyruvic acids. And then for each individual pyruvic acid, we are going to strip off 1 carbon dioxide and turn it into an acetyl-CoA that has 2 of those original starting carbons left. And then that acetyl-CoA is going to be entered into the cycle, and the very first molecule of the cycle is citric acid, hence the name, the citric acid cycle. So we started with 6 carbons in the glucose molecule. We then broke it down into pyruvic acid. Pyruvic acid has 3 carbons. We lost a carbon right here in the form of carbon dioxide. We're left with 2 of our starting, and then we dump it into the citric acid cycle, and we lose a carbon dioxide right here, and we lose a carbon dioxide right here. So for those 2 carbons that we dumped in, we then lose 2 carbons during the cycle in the form of carbon dioxide, and the cycle just keeps repeating over and over and over again. It's worth emphasizing that the citric acid cycle starts with citric acid, and then we have oxaloacetic acid as the ending molecule. This oxaloacetic acid combines with the acetyl-CoA in order to make the citric acid of the citric acid cycle. So for every glucose molecule that enters cellular respiration, we ultimately are going to lose a total of 2 carbons by making acetyl-CoA, and then 4 carbons in the citric acid cycle. Another way to think of this is that 1 glucose gets turned into 2 pyruvic acids, and then everything down here is multiplied by 2 because we have the 2 pyruvic acids. So when we think of the citric acid cycle, to bottom line it for you, our citric acid cycle is going to collect high energy electrons, and those high energy electrons are going to be carried away by NADH, or by NAD positives and FAD positives, in the form of NADH and FADH2s. And these high energy electrons are then going to be dumped into the electron transport chain. As we look at the citric acid cycle, it takes place in the mitochondrial matrix. To review, this is the liquid on the innermost part of a mitochondrion. Mitochondrion is the singular form of mitochondria. And as we're looking at this mitochondrial matrix, we need to get the carbons from the pyruvic acid into the mitochondrial matrix. So we take pyruvic acid, when we turn it into acetyl-CoA, or acetyl coenzyme A, and that acetyl-CoA combines with the oxaloacetic acid to make the citric acid, like I talked about 2 slides ago. And then we have a bunch of chemical reactions that we will not talk about in detail. These chemical reactions eventually take the citric acid that we start with and turn it back into oxaloacetic acid. And during these reactions, we remove high energy electrons. Please, oh please, oh please, do not memorize everything on this slide. This is not a biochemistry class. We're going to talk about some of the highlights though. So we start with an acetyl-CoA carrying 2 of the starting carbons in, and that acetyl-CoA is combined with the oxaloacetic acid to make citric acid. And then as the cycle progresses, we lose our first high energy electrons right here. So there's our first high energy electron being carried away, and then the cycle keeps going, and oh hey, we lose a carbon dioxide right there. We lose another carbon dioxide right here. We lose another high energy electron right there. It's notable though that the citric acid cycle does generate 1 ATP for every rotation of the cycle. So down here, we make guanine triphosphate. And guanine triphosphate is so unstable. It's such an incredibly high energy carrier that guanine triphosphate can turn ADP into ATP. So we indirectly produce 1 ATP for every rotation of the citric acid cycle. We just have a guanine triphosphate as an intermediate. Here we produce an FADH2, so we carry away some more high energy electrons here and some more high energy electrons right there. Let me shift colors for you. In red, I'm going to circle the high energy electron carriers. So we have lots of high energy electron carriers. 4 carriers per rotation, 8 carriers per glucose molecule. And then per rotation, I'm going to circle them in green. We have a carbon dioxide. Oop, move this up here. Carbon dioxide right here. And we have I'm having a brain hiccup right now. Where did that other carbon dioxide go? Ah, right there. And then the other carbon dioxide right there. So we also release 2 carbon dioxides per rotation of the citric acid cycle. That's all we have for this recording on the citric acid cycle, or Krebs cycle, or tricarboxylic acid cycle. You can pick your poison, whatever floats your boat. If you have any questions or comments, please feel free to post them on the class discussion board or to shoot me an email. And as always, class, happy studies.